Hello. Today we are actually going to go back to biology to explain today's lesson. So, in biology, we started to learn about the cell for a long time, but we know that the knowledge that we gain from the cell didn't come from only one person. Now, I don't know if you remember all of those scientists, but in science, usually in big theories like this one, knowledge comes from the creative thinking and the innovation of the collaboration of many different scientists. Sometimes it takes a lot of years between one group and another one, but it usually knowledge builds upon itself. Just like this tower resembling Legos, this is actually a building in the Netherlands, and just like one Lego builds upon another one to build a tower, this is how it usually happens in science. Knowledge builds upon itself based on all of this creative thinking and knowledge and innovation of many scientists working together. So let's review that with the cell theory. Now, this is a typical cell. Maybe you have not seen this one in particularly, but you've seen so many of them that you can easily recognize them. We know that it is an eukaryotic cell because it has a nucleus, and we can easily see some of the other organelles, the cytoplasm, the cell membrane, and so on. So, just take a minute to think about the cell, and just think about how people used to think before they were able to observe a cell. What do you think people thought that living things were made out of? Do you think it was only one person who came up with cell theory? And just to review, cell theory states that all living things are composed of cells. Cells are the basic units of structure and function in living organisms, and new cells are produced from existing cells. So all of this knowledge didn't come from just one person. And to understand all of this, we have to go back to the 1600s, to the study of Robert Hooke. Hooke was a scientist who had great technical abilities to invent different ways of using the microscope. He would vary the amount of light, and he would constantly modify things in the microscope. So this is one example of the microscope that he used. And one day, he was actually observing a quark, and we know that quark comes from plants, and he actually observed something like this picture here on the left. He had noticed all of these empty spaces surrounded by walls. It reminded him of the rooms in a monastery, and in Latin we call them cella. So he coined the term cell to refer to all these empty spaces surrounded by walls. In 1665, he published his book called Micrographia, where he used his microscope with a 50 times magnification. So he could observe in detail how some insects and other organisms look like. Now this opened the world to knowledge that people didn't even see before, with all the detail and just looking at all the detail of these organisms. So his book became very, very popular. And just a few years later, Anton van Leeuwenhoek was working as well with a type of microscope, the one that we see on the right. It looks more like a magnifying glass. He used it as a microscope, So one afternoon in the summer of 1674, he went to a lake near his house, and you know how scientists are, they want to explore and analyze everything that they see. So he was taking samples of water, as we can see here in the picture, in the drawing. And to many people, it would just look like dirty water, but for him, he wanted to see what actually was in the water. So he observed it under his microscope, and he discovered that the water was full with small animal-looking organisms. Here is something that he drew on his book. And all of this, he called them animalcules, for little animals. And now we can recognize them as protists and bacteria, but back then, just seeing this small organisms in so much detail that we couldn't even see before in a drop of water. It was something that a lot of people became interested in about, just the use of microscopes. And it wasn't until more than 100 years later that Robert Brown was working with flowers. He was doing an experiment to see the reproduction of angiosperms, flowering plants, and fertilization in them, that he observed in using this specific microscope that the plants, the orchid cells, all of them had something in common. All of them had these little dots that he identified as the nucleus. He thought that those dots meant something. They were there with a purpose, and he was the first one to identify them as the nucleus. In this picture, we can also see this stomata, that if you remember, they open and close to exchange gases in plants. And after him, shortly after, there were two scientists, Matthias Sheldon and Theodor Schwann. They also contributed to the cell theory, both of them working independently in different labs, but they used to share and collaborate with each other. So Schwann, his name might be familiar to you, as the Schwann cells in the brain. He was working with brain tissue and muscle tissue in his lab, and he noticed that all of the cells in that tissue had something in common. He noticed that they all had nucleus and cell membranes, and that was interesting to him. And a year later, Matthias Sheldon was working with plant tissue, and he realized that all the plant tissue that he was working with had also nucleus and cell membranes. So they worked together, they shared the information, and in 1839 they came up with what is now called the cell theory, stating that all living things are composed of cells and cell products, but they didn't know how this actually happened. They didn't know the mechanism of propagation until a few years later. Rudolf Virchow was the one who incorporated some stainings in the slide of the microscope. So he was using fluorescent and absorbing dyes with his organisms, and he was able to see that every cell is derived from another cell. Now we call this mitosis. But his knowledge, with the knowledge that has been incorporated before him, made possible our understanding of the cell theory, with the 3 points: that every cell is derived from cells, living organisms are made out of cells, and that cells are the basic unit of life. After him, there were other scientists who also used other kinds of microscopes and developed better ones to see all the organelles in specific of the cells and DNA and all the understanding that we gained so far. So, why do we use cell theory for a chemistry class? Well, now I want you to investigate and present the development of atomic theory. In chemistry, we're going to be talking about chemical reactions, physical changes using atoms. But our understanding of the atom has also changed over time. And as you're investigating this theory, I want you to think about certain questions. So, what are the parts of an atom, and who discovered them? How has the idea of the atom changed over time? What experiments were done in order to arrive to the current model of the atom? Do you think that there could be a new model of the atom in the future? So you're going to be incorporating all these ideas in your research, and then you are going to present your product using a software for a timeline, or using a presentation, or if you like to write, you can write newspaper articles from different time periods and compile that into a newspaper. Or if you have any ideas, you can discuss them with me. So I'm going to give you a rubric on how I want you to do this project. But I want you to start thinking about how science and ideas of science change over time as new experiments and discoveries are made possible from all of the scientists from different time periods, and how this knowledge changes. And maybe it could still change. I don't know. So if you have any questions, we're going to talk about this in class, and these are some of my references. Thank you.